So hello friends, welcome to Medicine 101. Today we'll talk about the drugs that we use in case of the HIV. So anti-HIV drugs. Okay. So before going to the drugs, let's know a little bit about the mechanisms, how they are going from the outside or the external environment within the host cells, and how they are fusing within the host cells, so within the genetic materials. Now, first we'll talk about here. Okay. Now see, I have already written all these things because it will take less time. Otherwise, the video will be long over here. Now see that the drugs used in HIV. Now HIV, there is a nature for the HIV that would love to react with the CD4 positive cells. Now from the terms, it's very easy to understand. HIV means human immunodeficiency virus. That means they will suppress the immune system. They will repress the immune systems. Okay, as a result, we will see a T lymphocyte count. Okay, that will be less than 250. Okay, that will be less than 250. Now, see in these cases that for example, is the virus is the CD4 positive T cell. Okay. Now within the virus, we know this the red one is the genetic material. That is the RNA. Okay, that is the over here. That is the what RNA over here. Now, this RNA virus. Okay. First, how they will fuse within the what CD4 positive T cell. That's the main thing over here. Okay. Now this is the capsomere. This is the capsomere over here. Now within the capsomere, there is a presence of one type of the surface protein that is known as glycoprotein 41. Now this glycoprotein 41, okay, that is having the receptor over here, that is the CD4 positive receptors, okay, the CD4 receptors over here. Now this glycoprotein, okay, 41, that will get bind with the CD4 receptor by a tunnel, or we can say on top of the co-receptor that is known as CCR5 or the chemokine receptor 5. Now see, you can see over here, this is the tunnel over here, you can see. Okay, that will form this tunnel. Now it will be easier for the genetic material of the viruses to get transferred from here to the host cell. Now we will get the RNA within the host cell. Whenever this RNA virus will come over here, the genetic material RNA we are getting over here. There is presence of such enzyme within the virus that is known as reverse transcriptase. Now the enzyme will convert the RNA virus into the DNA. Okay, the genetic material RNA into the DNA. Now, whenever the DNA will get formed over here by the action of the reverse transcriptase, the main problem we will see over here that there is presence of one type of the enzyme that is known as integrase. Now, whatever the problem, for example, the first part is the host DNA or human DNA. Now, they are having the capacity to get attached with the host DNA or the human DNA. If you just talk in terms of the human being over here, so they will get fused over here by the action of the integrase enzyme. Now after that, we know the rest of the process translation, they will produce the proteins. Now proteins means whatever the proteins. Now to produce a new virus just like this, they need to produce the different types of the enzymatic proteins, the surface proteins just like this, the capsid. Okay. Now we will see these proteins will be found first. Okay, but initially we will see these proteins will be inactive. Okay. Now to activate the proteins or just to get activated, these proteins forms. There is presence of one type of the enzyme that is known as protease. Now the protease that will convert this inactive protein into the active proteins. Now all the structures are over here. Now there is no problem to form the new virus. Now the new virus has been formed. Now it will be easier for the viruses to get out, and that will infect another healthy CD4 positive T cells. Just doing like this, they will just decrease the amount of the T cells. As a result, we will see the immune suppression. Okay. Now think about this thing, just to inhibit the growth of the HIV or just to kill them, we can use the different types of the drugs. Okay, now whatever the drugs we can use, till today we have it's possible to discover till today that the vaccine over here, that's use some types of the drugs for it. The first thing, we can just inhibit the fusions over here. Okay, if we use some drug that will inhibit the fusions of the RNA or we can say the HIV virus, okay, within the along with the the CD4 positive T cells. The second type of the drugs you can use that will inhibit the reverse transcriptase enzyme. Whenever this enzyme will be inhibited, as a result, this RNA will not get converted into DNA. So the further no process will be seen. Now the same thing is over here, that if we inhibit the integrase enzyme over here, then this DNA, the RNA is getting converted into DNA. Now this DNA will be not able to get attached with the host DNA. Further this process, and lastly we can inhibit the protease enzyme over here. Now the active proteins will be not formed, then there is no chances to form the new the viruses. So this type of the drugs we can use. Now today we will talk about two types of the drugs, the fusion inhibitors and the reverse transcriptase inhibitor over here. Okay, reverse transcriptase inhibitor and the fusion inhibitors. The first we will talk about first the fusion inhibitor. Now see, in the fusion inhibitor we can use the drugs in the different stages. First stage is over here. We can inhibit this GP41 over here first. So we can inhibit the GP41. Okay. The second thing, we can inhibit also this tunnel. We can inhibit the CCR5. So we can inhibit also the CCR5. And the last thing, we can inhibit also that what? This CD4 receptor. We can inhibit also this one. So in the three stages we can inhibit by doing the fusion inhibitions. Okay. The first thing, the first drugs we can use by using, okay, Infi. Okay, see it over here. Infivirtide. Oh, here if we use, then it is injectable. Okay, you can give it by injections whenever we will use this drug. So definitely they will inhibit the GB41. As a result, the further process will be not seen. Okay. The second thing we can also use here over here, Maraviroc. Yes. So if we use the Maraviroc, then Maraviroc will not let, okay, to form the tunnel. Whenever this tunnel will be not formed, then there is no chances to get transfer of the RNA within the host cells or within the CD4 positive T cells over here. Okay. Then the third thing, we can also inhibit this CD4 receptors over here. Here we can use the evolizumab. So whenever we will use the evolizumab, okay, it is also injectable. We can use this type of the drug over here. Maraviroc we can give orally, okay, and this one we can give by the injections as well. Now the second type of the group of the drugs, we can inhibit the reverse transcriptase enzymes. Now if you want to inhibit the reverse transcriptase enzyme over here, now we can inhibit in the two ways. One is competitive, another is non-competitive. I have written over here, okay. So within the competitive, we will get two types of thing. One is nucleoside, one is nucleotide. From the aspect of the nucleoside, we can use some drugs just like the zidovudine. We can use the lamivudine. We can use the stavudine. We can use the dodanosine. Okay. Along with that, we can also use with respect to the nucleoside, we can also use the avacavir. Okay. So now if we can also use in terms of the myocardial infections this avacavir. Okay. If someone is having myocardial infection, if the patient is getting affected with HIV, you can use this drug. Now the side effect for this type of the drug we can get over here, that it can cause the pancreatitis and the peripheral neuropathy. Okay, the pancreatitis and peripheral neuropathy. Among these drugs, the dilanosine is having the most chances to cause the pancreatic or the pancreatitis. So dilanosine is having the maximum chance to cause the pancreatitis among these all drugs. Okay, nucleosides. And in terms of the nucleotide, we can use the tenofovir. From the T terms, you can remember T for nucleotide, T for tenofovir. Here you can use this drug. Now among these, the first line drugs over here, that means in terms of the nucleoside, that the safest one is the lamivudine. So lamivudine you can use over here. Now if you talk about in terms of the non-competitive inhibitor over here, we can use in the two groups, the first group and the second group. Now if you talk about in terms of the first group, what type of the drugs you can use, the most important one is over here, the mid one, nevirapine. Now nevirapine, it is used to prevent the HIV from placenta to, from mother to, we can write from mother to baby. From mother to baby, it will prevent this transmissions over here. Okay. We can also use the dilaviridine, we can also use the efavirenz. Okay, we can use this one of the drugs as well. In the second group, we can also use the rilpivirine or rilpivirine. Just try to remember. Atravirine, we can use the doravirine, same. The virine is the precursor. So atravirine, rilpivirine, we can use the doravirine as well. So this was all about the drugs in the first two drugs, the fusion inhibitions and the reverse transcriptase inhibitions. Okay, so first the same thing, the reverse transcriptase that will be inhibited and the fusions inhibitions over here. So if you do like this video, please don't forget to subscribe my channels, and thanks for watching, and please share for your friends as well. Okay, thank you.